that they produce. And then we're going to look at how the hormones actually affect the lives of individual bees, and how the hormones also affect the colony as this superorganism, this big social group that they live in. So there are three systems in the honeybee. A nervous system, which I think we're probably all aware of. You know, if you just squeeze a bee accidentally, you know that the nervous response is to sting you, unfortunately. So that's the nervous system. Then you have an exocrine system. Now, Claire, exocrine, that suggests pheromones, yes?
Yeah, it's the glands which open onto the open of the body, onto the outside of the body, and pheromones are chemicals which affect other organisms of the same species.
Okay. Now, the endocrine system would suggest that it's an internal system. And so it's this internal system whereby the body produces chemicals, often in the brain, and those chemicals actually act on other tissues within the body to create a reaction. So if you're looking for a definition of what a hormone actually is, it would probably be something like substances that are produced by specific cells that are physiologically active and that produce their effect inside the organism. And we're going to try and...
Yeah, so basically, Martin, you're saying chemicals which send messages inside the body rather than outside the body.
Yeah, so they're not influencing particularly other bees around them. The activity is being confined to the bee itself. So it's how the bee itself reacts rather than effect it has on other bees in the colony. But just to compare that with what happens in the humans, or indeed some mammals in general, higher creatures. So if you think about human beings here, we've got an endocrine glandular system, and you can see them marked up there in the orange there. So you've got specific glands in particular areas spread right through the body. The pituitary, thyroid, pancreas, the ones in the naughty parts down the bottom there. But the thing is that these glands are quite specific into where they are, but they actually secrete the hormones into the blood supply. And if you think of the blood supply like a ducted pipe network around the body, so the gland is able to deliver the hormone, the chemical, into this pipe system, and then it's delivered to where the receptor cells are. But if you think about that in difference to a honeybee doesn't have a piped blood system. It has just a lot of hemolymph in the abdominal cavity there, and it's just sloshing around in there. So any chemical that's delivered by the endocrine system basically goes to everything, and it's only the specific cells that require it that pick it up. So it's a much more open system than you would find in a mammal, for example.
Does that make, Martin, does that make it a bit more hit and miss? Does it make it less specific?
It can do, and it can mean that certain ways that hormones perhaps react in humans, whereas, for example, a hormone may react in a male different than it will react in a female, because it can be targeted to the specific area of a male or a female where it's required. In a honeybee, that hormone is just sloshed to everything, and therefore it's not so specific. So if we compare what we see there with our mammal there, this is a stylized version of what you would get in an insect. I couldn't find a picture of a bee, so you've got, I think it looks like a cockroach or something like that down the bottom. But if you look at all what we see on the screen there, is actually just confined to the head area in the insect. So whereas the mammals had glands all the way through the body, in the insect, it's mainly and virtually completely in the head of the insect. And so you have these neurosecretory cells that are in the brain. And then following on from those, you have the corpora cardiaca. After that, the corpora allata. And then finally, the prothoracic glands, which are slightly different, and we'll come to those through the talk. But can you see that in actual fact, in the insect, in the honeybee, all these glands sort of in a way morph into one. They're all sort of joined together, and all you have are like swellings in the tissues at that particular area, rather than in the mammal you have defined glands. This is much more all sort of joined up, and there's linkage between all the glands. So just a little bit of background about how we got to this stage, and we've got our rooster on the screen there. And if you look at the history behind hormones, you'll find that in actual fact, it's quite a new science. So up in about 1849, it's said that they actually did some experiments on chickens. And I have to say, I'm always a little bit sort of worried about these types of experiments. They make me cringe a little bit. But what they found is that if you castrated a male chicken, then it ceased to be a cockerel. It lost those cockerel-like tendencies. And then if you implanted the testes from another cockerel back into it, suddenly it started to become a cockerel again. And so it was decided that there must be some sort of secretion from the testes in this creature that was making it a cockerel rather than a neutered bird. But it wasn't until the early 1900s that the idea is that you had a chemical message as a hormone being used to provide these alterations in the body. Now, when it comes to honeybees, most of the work has been done in the 1900s, and it was done particularly on the molting hormones. And we'll look a lot more in detail at these molting hormones as we go through the talk tonight. But you can see that the activation hormone was first discovered by Kopek in 1917. And he found that in actual fact, these hormones were more to do with neurosecretory cells, which remember are found in the brain, rather than gonadal tissues like you would find in that rooster. So where, again, rather than being a secretion from a specific body within the creature, it's coming from this more general area. So it's a different system, a different style. And then juvenile hormone, and we're going to talk a lot about juvenile hormone tonight, and the molting hormone, they came along a little later, but they're all interesting, and we're going to see how they all sort of work together, and one may affect another one as we go through the talk. So as we said earlier, it's a very complicated system, and it's quite vast in how it affects the life of the honeybee. But we're going to concentrate tonight on four particular areas: larval development, age polyethism, caste determination, and the mobilization of sugars within the body of the honeybee. So just to remind you about the four key parts of the endocrine system and what we're going to talk about: the neurosecretory cells, which are found in the brain, the corpora cardiaca, just a bit further down, and sometimes they receive secretions from the neurosecretory cells and then pass them on to other tissues. The corpora allata, that particularly is responsible for the production of juvenile hormone. And then the prothoracic glands, which are only found in the larva because they are to do with the molting process. Okay, so the ones we're going to start with first are the neurosecretory cells. And the neurosecretory cells produce neurohormones, and they control many of the behaviors which help to make the superorganism what it is, what makes a colony function as a colony. Now, a lot of books will tell you that they're actually not true hormones in the full sense of the word. Do you remember our definition at the start? We said that a hormone is a chemical that is secreted from the brain and then operates elsewhere in the body. But the neurosecretory cells and the neurohormones, they actually are produced in the brain and they act in the brain. So they target various cells and they target behaviors. They're much more about behaviors than the other hormones. And so we're going to look at how these neurohormones affect the behaviors of the bee. And I'm going to pass this one to Claire because I know she's really interested in this. And because not only that, you know, these neurohormones that the bees have are actually very, very similar, if not the same, as the type of neurohormones that we ourselves have. So, Claire, I'm going to pass this to you.
Yeah, we do share quite a lot of the what I call the neurotransmitter hormones. As honeybees, we share dopamine, we share serotonin, a kind of adrenaline that the bees have, and dopamine in people is associated with risk-taking behavior. So people with high dopamine levels like doing things like bungee jumping or even gambling. They're risk takers and they're explorers and things. But in the honeybee world, bees that have got higher dopamine levels are the scout bees. They have got seek and find behavior, and it's something to do with the way they were when they're in the cells. Perhaps they were incubated at a slightly higher temperature, a bit of epigenetics, which made the dopamine levels higher. But it's seek and find behavior. And this is true for nectar or flower foragers, but also scout bees, which go out and search for new homes. You know, when a swarm goes out and the scout bees go out and search for a new place to live. And didn't you say, Martin, earlier, we were looking at some research on this, that drones have higher dopamine levels just before they go out on their mating flights?
Yeah, that's what they think. You know, these drones have to fly out to drone congregation areas where they're going to meet up and wait for a virgin queen to come through. In order to make this journey again, that they seem to have increased dopamine levels, so increased neurosecretory cell activity, sort of late morning, early afternoon when they're going to make these mating flights. So it appears to encourage them to go out and be taking their chance, as it were, in the
Daring.
bigger.
It's a daring. It's a daring hormone.
Yeah. Now, what about serotonin then, Claire? We've got serotonin as well as humans.
Yeah, serotonin in people is associated with being happy. You know, if you've got high serotonin levels, then you're able to be content. And people that with low serotonin levels sometimes suffer from depression. So there are lots of drugs that will called SSLRs, or it's to inhibit serotonin going from the brain into the body and doing other things. So it tries to keep the serotonin in the brain. And we all know, us ladies, that chocolate will increase our serotonin levels just as much as anything else. And so it seems to be a hormone which levels responsiveness to different things. And in bees, it's slightly different, isn't it? It's responsiveness to alarm pheromones. So it's increasing your response to something.
Okay, and the other one that we share particularly with humans, octopamine, Claire.
I don't know. Do we have octopamine?
Well, I thought we did, but in terms of a bee colony, it's certainly one of the things that kicks in when bees come under heat stress. So, for example, when they need to start cooling the hive, I don't think that's going to be happening anytime soon, the weather we're having at the moment, but no doubt we will have a hot spell at some stage through the season. But it's those bees that have lower levels of octopamine that seem to start the thermoregulation more. So it's all
it's all about thresholds.
It is all about thresholds. And what is really interesting is the fact that the levels of neural hormones, so in other words, how responsive these bees are to certain circumstances, seems to be very much related to the levels and genetics of the queen. So the fitness of the queen, how well mated she was. And I know, Claire, you're always on about that well mated queens produce better colonies.
Yeah, so a queen that's managed, it's not just the amount of drones that she mated with when she went out on her mating flight or occasionally her mating flights, it's the diversity of the drones that she mated with. So a queen that's managed to mate with as many unrelated drones as possible will have not only better pheromone levels and better gut bacteria, but she will also have, or better gut microbiota is the new way of saying it, but she also has better hormonal things. So that you know, a well-mated queen, a queen that's managed a really diverse mating, will be fit on every level, and she'll inspire fitness with her colony. It's not just the number of patrolines in the colony or the subfamilies, it's all of this being able to respond to changing thresholds, the ability to be able to do that.
So let's have a look at these prothoracic glands. Now, I have to say, when we were researching this, I had terrible trouble because some books refer to prothoracic glands and other books were referring to ecdysal glands. And I was getting very confused trying to research these two different glands. And then it dawned on me that in actual fact, they're one and the same thing. So some people call these glands prothoracic and others call them ecdysal. But that basically what they do is they secrete hormones which influence the molting of the larva. And so they're only found in the larva. There's no need for the adult bee to molt, and therefore they don't have these glands. So when the pupa, the larva turns to a pupa and the pupa does that final molt to become an adult, these glands are sort of broken down and the tissues reprocessed, so they don't appear in the adult. Okay, but how they actually work in the larval molting, I think, is really interesting. Claire says,
how many times does she molt?
Okay, so when the egg is laid, that takes 3 days for the egg to hatch. Yes, and then from the hatching through to when the larva is sealed over in the cell, that's about 6 days. And in that 6 days, the larva undergoes 4 molts for when it's sealed. And then after it's sealed, it undergoes a molt to become a pupa, and then a further molt to become an adult. So 6 in total, 4 in the open cell and 2 when the cell is sealed. No.
And when they can I just a question. So they molt because the skin won't stretch anymore?
Yeah, these larvae are growing really quickly through that 6-day period. If you think about it, the first day you can hardly see them, and by day 9, 6 days later, you know that they're effectively filling the cell up. So they're really rapid period of growth, and so each time that the skin becomes stretched on the larva, it's going to send a signal to the brain saying it's getting a bit tight in here, and so the molting process starts. And overall, the molting takes about 30 minutes each time. So for 30 minutes, every sort of day and a quarter, day and a half, the larva is going through this molt. Now, I have to say, I've never actually seen a larva molting. I don't know whether anybody has or whether you have, Claire, but it does seem to involve quite a bit of wriggling from what I gather. You know, the skin splits and then the larva sort of forces its way out through, and the dead and old skin sort of gets incorporated into the structure of the cell, and then the new fresh skin, which is very stretchy and can take up the bigger body size, that sort of firms up, and so the molt takes place. But of course, the interesting thing is, as we said there, sometimes the larva wants to molt but still stay a larva, And sometimes it needs to change from a larva to a pupa, or from a pupa to an adult. And that's what we're going to look at. How do they do that? How do they know whether they want to stay a larva, or how do they, or they're going to turn into a pupa? And that's under hormonal control. So the neurosecretory cells in the brain, they release this thing called prothoracic tropic hormone. We'll call it PTTH. And this stimulates the prothoracic or the ecdysal glands, whichever you want to call them, to release the molting hormone ecdysone. But like we've said, the larva needs to undergo different types of molt. And so which type of molt it's going to go through will depend upon the interaction with another hormone, juvenile hormone. And we're going to talk a lot about juvenile hormone through the talk. So we're going to have two hormones working together to control the type of molt that the larva is going to go through. Now, juvenile hormone is secreted in the corpora allata, and that's just secreted. Let's see if I can get a laser pointer on here a minute. There we go. So there they are there, those two brown objects, okay, look like tonsils actually, but there they are. That's where
they look like kiwi fruit.
Yeah, okay, kiwi fruit, I'm cool with that. So they're present in the larva and the adult. We know that that they're present in both stages, but this is a big but. The way that the juvenile hormone affects a larva is different than the way the juvenile hormone affects the adult. And we're going to take the two things in turn. So we're still going to concentrate on the production of juvenile hormone by the corpora allata in the larva. Okay. So this is just a breakdown basically of what happens. And we start here. Here's our egg at day 1, goes to 3 days and it hatches. Then we have an first instar, second instar. What does instar mean, Claire?
I just think it's a reference to a time scheme.
Oh, okay. So 3rd, 4th, 5th. Then we get final stage larva. Then this is not, interestingly, that this is not a molt here. This prepupa does not require a molt. It goes from larva through this stage and molts to become the pupa. This is sort of an in-betweeny stage, but it doesn't require a molt. And then it goes through here, the final stage pupa, and then it finally becomes the adult. So it's the juvenile hormone that's responsible for the maintenance of larval structure, and it inhibits the metamorphosis. So what we're really saying is that juvenile hormone in this instance is doing exactly what it says. It's keeping the larva young. And so when you have high levels of juvenile hormone, the larva will continue to be a larva even though it's done a molt. But as the amount of juvenile hormone lowers, so the pupa, the larva will age as it does its molt to either become a pupa or an adult. So what we've tried to do is just put it in a little sort of flow chart here to see if that makes sense. So here we have our neurosecretory cells up in the brain. And they are going to secrete the neurohormone prothoracic atrophic hormone. And they stimulate the ecdysal glands to secrete ecdysone, the molting hormone. So, okay, so we've got our pathway there. And then once we've got our ecdysone, that produces a larval molt. But at the same time, running alongside this, we have the corpora allata that is secreting juvenile hormone that determines the type of molt. Okay.
So is the corpora allata a bit of fine tuning?
Yeah, it doesn't instigate the molting, but it determines the outcome. So,
instar means, by the way, it means just a developmental stage.
Oh, okay. You've looked that up, haven't you?
Yeah.
Yeah.
I Googled it.
Okay. So initially, just to read what it says on the screen there, initially the larva has these high levels of juvenile hormone in the hemolymph, And so the first 4 molts are able to be larva to larva. But however, as the level of juvenile hormone drops, so the next molt after the cell is sealed becomes a larva to a pupa. And at the time of the final molt, juvenile hormone is completely absent. It's all gone. It's all been used up. And so the molt then is able to be from a pupa to an adult. So it's this level of juvenile hormone that determines how the outcome of the molt is going to go. The other interesting thing I thought, well, I think these things are interesting anyway. You pay your money, you take your choice. But can you see when we see these pupa in the cell, they're white, and then we know that the adults, just as they emerge, become brown, and that's also under hormonal control. It's under the hormone called bursicon. I don't know much more about it than that, but it just shows that every aspect of the honeybee's development is controlled by a hormone. So even the coloration of the bee as it emerges.
You can just see the wings forming there as well.
Yeah, you would do. And sac brood, Claire. We all get a bit of sac brood, yes?
Well, speak for yourself. You generally it's difficult to look at a hive that hasn't got a little bit of sac brood sometimes. And sac brood is a failure of ecdysing. The larvae can't molt.
Yeah, it's basically the virus.
The virus which must affect the hormone.
Yeah, the sac brood virus affects the ecdysal glands. So although the prothoracic glands may be secreting the activation hormone, in actual fact, the ecdysal glands don't produce the ecdysone, the actual molting hormone. So that the sac brood is cutting off the pathway, the hormonal pathway within the larva. So that's how juvenile hormone works within the larval stage. But let's have a look at what happens in the adult bees. So it's important to remember that although you have a corpora alata in the larva that produces juvenile hormone, and you have a corpora alata that produces juvenile hormone in the adult, they are not the same gland. The protein origami, as Claire likes to call it, at the metamorphosis that happens between the larva and the adult, the gland gets broken down. The corpora alata is broken down and it is reformed. Now, it's reformed in a similar fashion and it produces the same hormone, but the hormone is going to work in a different way. So you mustn't confuse the two. So juvenile hormone, again, as I say, secreted by the corpora alata, works inversely with this phosphoglycoprotein called vitellogenin, which is produced in the fat body. And Claire, some people actually say that vitellogenin is a type of hormone in itself, don't they?
It can have a hormonal action. It actually has, it performs lots of functions within the honeybee, but it does have this relationship with juvenile hormone, like a sort of, as juvenile hormone goes up, vitellogenin goes down and vice versa. So there is a relationship between the levels of vitellogenin and the levels of juvenile hormone.
Yeah, so if you think about it, when we were talking about the larva, it was high levels of juvenile hormone kept the larva young. But in the adult bee, high levels of juvenile hormone are actually making the bee old. Is that right, Claire?
Yeah, when a bee's born, she builds up these levels of vitellogenin in her fat body. So young bees, when they're born, have these fat bodies, which are areas of fatty tissue just below the exoskeleton or just underneath the exoskeleton, particularly in the abdominal area. And these fat bodies produce this glycolipoprotein called vitellogenin, which then disperses as a way of dispersing that. It's somewhere between a protein, a sugar, and a lipid, isn't it? A fat, and it helps to disperse it into the hemolymph. But it has many different functions, so it can have a hormonal effect in that it will, to a certain extent, affect the lifespan and the sort of duties, the polyism of the bee. So, but as the nurse bees feed larvae, they use up their levels of vitellogenin. So they run out as they go through their life. They start running out of vitellogenin. So it's quite a complex thing, this vitellogenin.
Okay, so here we have a very inappropriate drone talking to a worker bee, and the drone's going to say, "You seem to look a bit older today." And the nurse bee saying, "Don't you start, my juvenile hormones kicked in." So there's even un-PC-ness even in a honeybee colony.
Un-PC-ness.
Yeah, that probably didn't come out right, did it? But hey ho. So do you want to just this fat body? Is this a fat bee on the left with a big fat body, or have I got it wrong here,
No,
Claire?
you won't see the bees that have got high fat. It's just a fatty tissue that's just so it doesn't make the bee look fat at all. It's just there underneath the exoskeleton, and they're well developed. And again, well-nourished larvae have these fat bodies, which then produce many things, but this substance called vitellogenin. And on the picture on the right-hand side, can you point that picture out, Martin? And go into the bee with that's a young bee with high levels of or high well-developed fat bodies. And again, those are the bees that will go into winter. Bees that have got go into the winter have these well-developed fat bodies. And then if we go to the other side, that's a forager bee. So she's used most of her fat bodies up. So her last job would be foraging. So foragers have very low levels of vitellogenin, whereas nurse bees, when they first start nursing, have very high levels, but it's something that get used up.
Okay. And the fat body also does other things. So it helps to maintain immunity against bacterial and fungal diseases. So if you have a fat body that's been damaged or in any way, or is not producing correctly, then that can leave it open to bacterial diseases. And one of the things we do know about EFB, European foul brood, is that it's very much related to poor nutrition, and poor nutrition can manifest itself in a poorly developed fat body tissue. It's also important for, like Claire said, lipid, carbohydrate, protein metabolism, stores nutrients, and that's quite important because this fat body is what's used to drive forward the glandular secretions in the body. So having good nutrition at the stage when the fat body's been produced is
in the
really
form of
important.
stuff like glycogen, which will give if you've got lots of glycogen, say at the base of the flight muscles, it gives them the energy to fly, and eventually that glycogen will run out as a bee reaches the end of her life. Her glycogen is used up, and that's really what is the end. That's the end of her life then.
Okay, so here we've got a young bee just being born, bless her. Here we go. And she's brimming with vitellogenin, Claire, yeah?
Well, she will build that up after she's born. So it'll be the first few days, but she'll build up her vitellogenin then.
Okay,
So
and then as she goes through her life doing all these jobs, this is going to use up.
Yeah, it will use it up. Yeah.
Okay, so let's just have a look at a few of these jobs. And I know when I was learning beekeeping, I was told that it was very strict and rigid, Claire, that the bee did this for this many days and this for this many days, all the way through her life. But I think these days we think it's a bit more loose than that.
Well, even if you read Winston, which was Mark Winston's Biology of the Honeybee, a bee's life is very flexible. So yeah, in the first few days of her life, she will probably do more cleaning, but she doesn't clean relentlessly. She might do 10 minutes cleaning a bit of a cell, and then she'll have a rest and do something else. And some of the jobs like nursing or producing food for the larvae, that takes a huge amount of energy. So she won't do that constantly for that time. She may do produce some larva food and feed the larvae, then she might have a rest, then she might do a bit of light cleaning, then she might have a rest, then she might do something else. So it's very much mixing match. They're very much multitaskers, but the kind of jobs she can do within the range depends on the glandular development. So it all depends on the needs of the colony as well. What is needed. If suddenly there's a field of rape comes out and they need lots of foragers, then some of the nurse bees will be fast-tracked to be foragers for a moment until that rape's all gone. So everything in the hive is really, really flexible, and that goes for the jobs. They don't go through this very strict sequence of jobs. And some bees will be specialists. They'll spend much longer on some jobs than others. So you might get bees that specialize in wax production, and they'll spend much longer doing wax production and then have a very short foraging life. So it's not quite as straightforward and simple as we were taught. It's much more complex and more fluid and flexible than that.
So just to recap, in the young bees, you have high levels of vitellogenin, which suppress the corpora alata from producing the juvenile hormone. But as the worker uses her glands to produce food and wax and things like this, her levels of vitellogenin fall. Therefore, the corpora alata is no longer suppressed, and the juvenile hormone secretion is allowed to increase. And as the juvenile hormone levels increase, so the aging process within the bee happens. Now, under normal circumstances, that would be something that would place over the 5 to 6 weeks of the age, the lifespan of a bee at this time of the year. But there is something that can affect this, and it didn't become apparent until just a few years ago. And this is the work that was done by Dr. Samuel Ramsey, and he did some groundbreaking work on the feeding habits of Varroa. And we have always been taught over the years that the Varroa mite feeds on the hemolymph, on the blood of the larva and the worker bees. But we've now been given to understand that by Samuel Ramsey's work, that they're actually feeding on the fat body tissue. And so that has huge implications for our bees. So, Claire, this riddle for you: when is a young bee not a young bee, but an old bee?
If a bee is heavily predated by Varroa, so if Varroa is feeding on the fat body of the bee and using up all of that, the ability to make vitellogenin, then what happens is that bee ages very quickly. So nurse bees or young bees become foragers well before their time. And that means all those important jobs in the hive don't get done. So the cleaning and the nursing of the larvae and the undertaking and the wax production, and bees become foragers. Now that colony will become very unbalanced, and they may go out and get loads of honey, but they probably won't go through the winter. And they can be very compromised. Those bees that go into the winter haven't got the fat bodies to be able to withstand a winter. They will die before the springtime, before the young bees are born to replace them. And so, although it's not, your colony can look perfectly fit and healthy going into winter, but if they've been heavily parasitized by Varroa, very often a lot of winter losses are not obviously Varroa. It's almost like a silent killer. They just haven't got the fat bodies to get through the winter, and the colony will probably fail early spring or about nowish because that colony's become so unbalanced.
Interesting. So, moving on to another subject, and it was part of the title: what makes queenly queens? Okay, so what role do hormones play in the production of queenly queens? So we know that all fertilized eggs can either become a worker or can become a queen. That they are one and the same, and it's more to do in the way that they are fed. But it's the feeding and the relationship between the hormones that are secreted as a result of that will make the difference. There she is. Okay. So here's what we're seeing. We're seeing quite a lot of this. Larvae sat in a pool of royal jelly, and it means that swarming's underway and queen larvae are being nourished. And we know that the larvae that are going to become queens are fed this royal jelly. It's this combination of the hypopharyngeal and the mandibular gland secretions. that are produced by the worker bees. And as you can see, and as we know, they're fed copiously. They're just floating around in it. But this royal jelly has a really high sugar content, about 34%. And the idea is that it encourages these larvae to eat much as possible. It stimulates them to eat, and they eat and eat and eat. And if you compare that with the sugar content of a worker larvae, that's only about 12%. So it's about a third of the amount of sugar. And so they don't eat so much. And it may be to do with size, but it's much more to do with, as we shall see, the production of hormone. There is one interesting thing, actually, and that is if you think about these larvae that are in these pools of royal jelly, that the queen cells always hang down, don't they, on the combs, and yet the larvae don't fall out. And equally, they don't sink in it either. They just get held on the surface, and it's this level of sugar together, and the pH and the overall consistency of the royal jelly is just at the right amount to keep those bees, those larvae, stuck on the surface of the jelly without either falling off or drowning in it. And it's a really interesting thing. I'd never really thought about it until we put the talk together.
If you give them a good shaking.
Well, they probably would do, yeah, if you agitated it really hard. But under normal circumstances, they just hang there and they're adhering to the surface. Very clever, I thought. So these developing queen larvae, they receive higher proportions of mandibular gland food than the workers do, and these contain high levels of pantothenic acid and biopterin than the worker larvae do. And so what happens is that the difference between the diet in a worker larvae and a queen larvae are noticeable at about 3 days after hatching. And anybody that does a lot of grafting will know that you've got to graft before this age, otherwise you're not going to get a good queen. And if you let colonies make emergency cells, if there's the queen's taken out, they want to convert larvae normally no later than 3 days. And it's this diet that they're receiving at this particular time is when the queen is going to be set up to be a good queen. This is when queenly queens are made, for want of a better way of putting it. And so what we know is that the levels of juvenile hormone, it's good old juvenile hormone again, are very important in the queens. And the juvenile hormone levels are about 10 times higher in queen larvae than they are in workers. And what happens is, I love this little sentence here. What happens is that the queen larvae is eating so much of this high sugary food that it causes midgut extension, and it causes a subsequent excitement of the stretch receptors in the gut. This tends to send a signal to the corpora allata, and this increases the juvenile hormone levels. So it's this high sugary food that causes this stretching and this excitement of these receptors that causes the corpora allata up in the head area to release more juvenile hormone and make more queenly queens. It's not only a question, though, Claire, of switching on certain genes, it's switching off some as well. Epigenetics.
Yeah, epigenetics is we've all got genes that can be switched off or on, and things, external factors can affect those. So the type of food that your mother ate while you're in the womb, whether she smoked or not, whether she took drugs, whether she had a good pregnancy or bad, those will all switch on and off different genes. It will affect our genetics, and so it is with the honeybee. And this copious feeding with royal jelly switches off some genes and switches on some genes, which will make the queen a different shape and able to develop ovaries and have the kind of physiology that she has.
Okay, this is just a little aside here. We all know that if you've got queen pheromone in a hive, then it actually becoming laying workers. We're all well aware of that. But in actual fact, what it is the fact that the queen pheromone actually suppresses the corpora allata activity in the workers, and so the workers don't become queenly, don't have high juvenile hormone levels because of the pheromone in the queen. So you've got a pheromone acting on other bees in the hive, and then within those workers themselves, the hormones being suppressed. So it's an interaction between those two systems, the exocrine and the endocrine systems. It's like with most things in beekeeping, it's not quite as simple as we like to think it is. Okay, you'll be pleased to know on to the last part of the talk now. And so my question is, what do they all have in common?
Can I say?
Yeah, go on then.
We found out about it recently, called tree hallows, which all of these, the brine shrimp, you can actually buy them in toy shops as they're a little sachet of powder. And when you add that sachet of powder to water, these little shrimps just come alive. They've been dormant for I don't know how long, but they start coming alive and they start flipping around in the water like little tiny translucent fishes. Or sea monkeys, and also plants that seem to be able to, you add water to a powder and these plants just appear. And this tree hallows is like a latent energy which can be activated by just adding moisture. So how does that apply to the honeybee, Martin?
Well, it's a bit of license here, but I couldn't resist it really. But the thing is that the most abundant sugar in the hemolymph in a honeybee is trehalose. It's not glucose, it's not sucrose or fructose, it's trehalose. And it's this idea that trehalose is an energy store. It's a reserve which can be called upon when it's required. And we'll see how important that is. For them.
Can I just say about Peter's eye drops?
Of course you can.
Yeah, because my partner has got dry eyes and he's got these eye drops. And when I looked on the ingredients, and it actually said on the label, 3% trehalose. It actually is used in the food industry as well. I've seen it in products for, when you're old and you can't absorb nutrients, like protein-rich drinks, vitamin drinks and things like that. It's in there. So it's this latent energy.
It also has another use in honeybees, particularly in the queen. The spermatheca in the queen has very high levels of trehalose. And just like Claire was saying about the dormant phase in the shrimps and the plant there, so the sperm in the spermatheca seem to be able to survive this dormancy period in the spermatheca because of these high levels of trehalose that are in place there. So it's such an interesting and complex sugar, but it seems to play a really important role in the honeybee life. So we've got three creatures on the screen there. We've got a cow munching some grass.
And
we've
dandelions.
Got a shopper and dandelions.
They do like dandelions.
And I have to apologise. I could not find a picture of a man going shopping anywhere. So it's a person going shopping, and then we've got a pride of lions sort of snoozing under the tree there. And I suggest, what actually motivates these three types of creatures there to either munch grass, go shopping, or go and hunt a wildebeest? And the answer is they're all motivated to forage because they're hungry. They're motivated by hunger. But what about our foraging bee? Is she motivated by hunger? Well, not really, because in actual fact, forager honeybees are fed before they leave the hive. So there's got to be something else motivating them other than hunger. They don't go out to collect because they're hungry. And what it appears to be is that juvenile hormone again seems to leave the nest earlier about as the bee ages, so the levels of juvenile hormone rise. So one of the rises. So one of the effects of this high levels of juvenile hormone is actually encourages or allows the bee to be comfortable to leave the hive to actually do this foraging and then to bring back food collectively, even though it's not hungry. So within this bee, you have this feedback mechanism whereby excess glucose can be synthesized and converted into trehalose or possibly glycogen. But basically what happens is that trehalose is a double glucose molecule. And so there's this feedback mechanism inside the bee that when the bee has excess glucose, it converts it into trehalose. And when it needs glucose, it breaks the trehalose down and mobilizes the glucose. So it's got this regulatory system going on inside the bee. And so what we need to think about are the decisions, as it were, maybe quite not the right word, but the decisions that bees collectively have to make about foraging, how they're going to fuel the flight to and from the nectar source. So in other words, there's no point in a honeybee foraging force going out to a nectar source that's not worth the effort to collect and bring back. There has to be a positive correlation between the nectar that they're going after and their ability to go and get it. That's Carl von Frisch, Claire.
It's matey in the leather shorts, isn't it?
It is indeed. And he did this really famous work on the dance language of honeybees. And we've now had a chance to look at it a lot more down the line. And it seems that the dance itself conveys much more subtle information than simply distance and direction. So what it's actually demonstrating to the other bees in the hive, the potential foragers and also the house bees, is the actual nectar source worth going after? And then what they determine is how much food they're going to need to get there. So I always thought, Claire, and we found out we're wrong, but the forager bees that were going to go out on this foraging trip, having been recruited, they just go to the combs and they just fill up on honey and go. Is that not right?
No, not at all. Obviously they can't go out, without any energy, and they need some stored energy because, bees will forage up to 3 miles sometimes, and flying takes an enormous amount of energy. So they need some fuel for their flight. But Martin found this out. I think it's absolutely fascinating. Not all those bees that are attending the dance are going to actually fly out to find the nectar. Some of those bees are picking up information about how far the others have to go, and they calculate just how much food to feed those foragers. They don't feed themselves. There'll be bees that work out how much food the foragers will need to get them to the source of the nectar or the pollen. So they will actually feed the foragers the exact amount of food to get there.
Okay,
is that right?
So yeah, so if you think it sort of makes sense when you think about it. If these forager bees just went to the honey stores in the hive, filled up their crop, this elastic part of the stomach, we'll talk about that in a minute, but if they just fill up their crop and then they fly out and use all this food getting there and then just bring back a load of wishy-washy nectar, well, it'd have been better to stay at home. So they actually only only actually carry enough food to get to the nectar source, and then they can fill up and then they come back again. So there's a positive gain from doing the flight.
Use your analogy.
Well, yeah. So rush down here, Newton habit. in South Devon than in Bristol. So you better have a round of sandwiches to get you there, young man. And then if you say, Mum, I'm going to Bristol, then she say, oh, you need a round of sandwiches
And a donut.
and a donut because you're going to take longer to get there. So it's that sort of analogy that the distance conveyed and the amount of food you're going to take to get there is how the workers are provisioned to get to this nectar source. And don't run away with the idea that bees are particularly efficient when it comes to flying. Their difference between when they're at rest and when they're flying, there is a 70 fold increase in the amount of energy that they're taking in. And if you compare that with a hummingbird or a small mammal or something like that, they only have a 7 to 14 fold increase in energy requirement. So the energy requirement for the bee is massive.
I think if you've ever taken film, and I took some film the other day, honeybees, and I put it on slow on my camera, and you watch them fly. Such inefficient flies. They're all over the shop. They sort of wobble everywhere. So they're beating their little wings and sometimes not getting very far because before they get any distance.
And the blood sugar levels in bees are some of the highest you find in the animal kingdom. And so what they found is that the concentration of sugar that these forager bees take out with them is about 30% sugar. Okay, and that seems to be enough to fuel their flight to the nectar source. So I just wanted to particularly for any newer beekeepers there, just to explain a little bit about how this crop works. Claire, do you want to just say about this little valve that's at the end of the crop?
So basically, the proventriculus is like it's like a valve which will allow liquids through one way, but it won't allow them back through. So it's a sort of opening and closing mechanism, which also does a little bit of filtering. So it'll filter out pollen grains and things. But it's not like a door opening. It's more like a camera lens. You know, when you change the aperture on your camera and it all closes down in a circular way. It's more like that kind of thing. It goes smaller in like that and wider out there like that. And basically the crop is just a modified part of the esophagus. So the tube that comes from the mouth down to the tummy is called the esophagus, and the crop is just like a bulge in the esophagus. It's almost like a sort of filling. You know, it's a sort of, if they carried it outside their body, they're going to be spilling it everywhere. It's just like a little internal elastic sac.
So in this crop, for the journey out to the nectar source, they're going to be carrying a sufficient amount of a 30% sugar solution. And as the honeybee detects that it needs more energy to keep flying, so a hormone from the brain releases the proventriculus valve and allows some of this sugar to pass through into the digestive system to fuel the flight. Okay, so that's how that works. But the question is, what are they going to do to get home? Because there's no point in eating everything in the crop to get back again. Again, they've gained nothing. So this is where the trehalose comes in. And as we said, the trehalose is a double glucose molecule, and the corpora cardiaca releases a hormone called trehalase, and that breaks down the trehalose into 2 glucose molecules. So on the way back, the flight is powered by the breakdown of the trehalose under hormonal control. So that's how they're able to get there by taking some sugar, but come back, bring a full crop of nectar back to the hive. Now, there is one other really important adaptation here where this is vitally important, especially in the springtime or in the middle of summer, and that's water, Claire. Water for bees is vital.
Absolutely. They need water for all kinds of reasons, for cooling the colony, and also to work back any solid honey which has gone solid in the cells. Don't like cold water because it will kill them. And so if you want to encourage bees into a watering hole, then put it so that it warms up. And I like to put moss in mine because I like to suck it up. It's almost like giving them another way of warming the water up. They're not very good swimmers either. So, but water, if they're going out to collect water, they have the energy, they're giving the food enough to get to the water, but then they haven't got the fuel to get back necessarily, because water's not going to give them anything, is it?
No, so again, it's this ability to get back from the water source is going to require the breakdown of some of this trehalose sugar to fuel this flight back. So again, vitally important, particularly in the summertime and also in the springtime as well. So really, just to recap, the hormones are the internal body chemistry, but it's development and it's life. They help to shape the development and the quality of future queens, and they encourage and control the behaviors of individual bees that together allow the colony to act as a super organism. So, personally, I find hormones absolutely fascinating in relation to how it shapes the lifestyle of the bee and the superorganism researched, and we don't really understand very much about it, but I hope we've shed a little bit of light on it tonight, and I hope we haven't bored you too much. But
I think it's not important if you can't remember the details, because I'm not very good. Martin's brain works much better on that kind of stuff than me. But for me, it's just seeing how everything syncs up, how such a tiny little insect has got such a complex set of behaviors, and what mechanisms make those behaviors happen and interact. You've got the pheromones, which influence hormones, and the hormones influence the physiology and the behavior of the bee, and they're all interreacting. And it's like the inside of our bodies, the liver affects the pancreas, affects the bile, affects, everything affects everything else, and you can't ever take anything in isolation. And it's just that I find fascinating. I can't remember all what individual hormones do what and where they come from, but I can get the gist of the importance of it.
Okay, so we'll stop the share, and I can't promise to answer any questions, but we'll do our best if anybody's got some.
Has everyone gone home?
We can't hear anybody. I don't know whether we've bored you all stupid.
So unmute.
We can't hear you, Liz. You'll have to unmute yourself. That's it.
Can you hear me now?
Do you want me to?
Yeah, we can hear you.
Right, listen to Mark.
Right. You talked early on about dopamine being increased in drones that were going out on the mating flight, to the mating zones. Is that all the drones in that particular colony will all together get an increase in dopamine, or is it like age related, so that the older drones within a colony will crank up the dopamine, but the younger ones probably won't?
Yeah, I think that's probably right.
Yeah, I think so. That there does appear to be that the hormonal production does seem to wax and wane through the day, and so it certainly seems to heighten at the drones that are at the right age to do mating flights, to be able to go out.
It's
and
12 days before they're sexually mature, isn't it? So it's obviously not going to affect the younger ones. Only when it's only the ones that have, like, gone into
up for it.
Well, no, what do you call it? Puberty.
Yeah. But there's an interesting analogy with that, with the dopamine in the drones and the queen mandibular pheromone in the virgin queens, because what they found is when the virgin queen goes on her mating flight, the level of her queemandibular pheromone spikes at about 2 o'clock in the afternoon when she's actually going on this flight. So it would appear that there's a similar mechanism. In one, it's a pheromone, but in the other one, it's a hormone. But there's a similar sort of mechanism that's readying the creature to do this mating flight.
Right.
Any more questions?
Yeah, I've got the one.
Come on then.
Find it.
He's just finding his question.
Okay, that's all right.
Oh, here we are. Yeah, you're talking about this process where the juvenile hormone restricts the metamorphosis. Is that all insects, or are we talking specifically only about honeybees here?
No, in actual fact, we've got a really good book called Insect Hormones. It's virtually unreadable to me. I'm just not clever enough to understand it all. But basically, the effect of juvenile hormone on the larval development is quite common across the insect world. And the juvenile hormone is important in lots of different insects, not just honeybees.
Right. Thank you.
Can I just ask a quick question? Do you think there's any impact, or has anyone done any research on climate change and neonics and how that might be affecting the animal plants?
I think it does affect them. Certainly. I just read a little tiny bit before we started to talk about the honeybee microbiome. The gut bacteria are very much the gut bacteria very much influences the hormone production. And so if you have poor gut bacteria, which would be influenced by pesticides and monocultures and not having a very diverse diet, then that would affect the hormonal responses. So yeah, you can't count those at all.
And do you think there's anything we should be giving our bees to enhance their bacteria?
I think the most important thing is diversity in diet. So we should all be planting for honeybees, not just our bees, but all bees. And it's the same with us. There's a whole lot of stuff in the media at the moment about our gut bacteria and how eating more than 30 different plants a week improves our gut bacteria. It's exactly the same for honeybees. They have to have a diverse diet, not just to get the whole range of nutrients that they need, but to get that gut bacteria healthy. And a field rape is not good for honeybees, but if you've got hedgerows in between with other plants around and other, wild field margins, then obviously the bees will pick and choose. They'll get the majority from the rape, but they'll also be, there'll be other bees feeding on other things. So that, not naturally, monoculture is not good.
Yeah, the other thing is that there are a number of amino acids. I think it's 20, but somebody that's done some modules may correct me. But I think it's about 20 essential amino acids that can't be synthesized by the bee themselves. They have to be able to bring it in the shape and form of pollen. And so, like Claire said, diversity of pollen is really important. And it's also the reason why, as you go through the season and you look at the frames with the pollen on, you get such a wide variety of colors, beautiful colors of pollen. They could probably get it all from one source, but they actively seem to seek out these diverse pollens and bring them back and use them. And so I'm sure that there is much more to that than just randomness. I'm sure there's
you'll get predominantly, say, dandelion pollen or pollen that's coming in at the moment. Probably you get lots of horse chestnut nectar coming in because it's very high sucrose content, but you just see little tiny bits of the brick red pollen and late pollen. They don't bring much of it in, but it's there all the same, little tiny bits of it, and those little tiny bits are important.
Right, is that no more questions?
Yeah, one more.
Go on then.
For winter feeding, Is there anything else that we can help them out to get all this? Is the sugar water enough for that? Or that's all the day done now, you can't help them.
Sorry, did you hear that, Claire?
I don't think it's here. Is there anything that we can put in the sugar water that we feed to build them up over winter that will give them some of these amino acids, etc., all these nutrients?
The amino acids would come via a pollen substitute, possibly, certainly not through a sugar carbohydrate feed. I tend to say to people that a pollen substitute over the winter is perhaps a bit of a waste, and it tends to be much more important in the spring, where you in an area where you don't get a lot of good natural pollen. I think it's like Claire said, it's basically down to good diversity of forage in the area. And if you happen as a group to have an area where you can influence the diversity of the forage and the pollen that's available, I think you'd be doing your bees a great service to try and plant that.
The other thing is, don't overwinter your bees just on sugar syrup. You know, try and leave them some of their own honey. That is much better for them to overwinter on their own honey if you can. You may have to supplement it, and we do. We have to supplement with what do we use? Belgo souk. But when we take the cappings off the honey, when we spin our honey out, we save all the cappings, and around about November we give those cappings back to the bees. So the more of their own honey they can overwinter on, rather than just sugar syrup. And I know most beekeepers try not to take all of the honey and just feed them, but I have been with beekeepers in the past year at the Abbey that they took every ounce of the bees' honey and they gave them sugar syrup. That's not very good at all. So try and find ways of letting the bees overwinter on as much of their own honey as you can.
Right, I think that's it, is it? Well, thank you very much. I think that's all the questions we've got at the moment. It's very
That's
informative.
okay, yeah.
It was a very informative talk. I know I've learned a lot. So some of these new beekeepers will, some of it might have gone over their heads because it's gone in at one ear of mine and out the other. But it was very interesting, and I thank you very much for all that extra work that you've done to get this to us. What else?
That's not a problem at all. Are you having cake there at the moment? It looks like I see people eating cake.
I think that one of the most interesting things for me is we're a very different genome with them, and a lot of the same neurotransmitters like serotonin and dopamine we have as well. You know, we all came from the same source originally, and although we have diverged quite a long way, there's still, we use hormones. You know, us women particularly, we know how hormones affect us, and the men probably by proxy. And although queen bees don't have a menopause as such, it is interesting to see what we do share.
Yeah, very interesting.
Thank you.
Well, thank you again. Send us an invoice and we'll pay you now.
That's okay.
Thanks very much, Martin and Claire.
It's all right. That's okay. Yeah, lovely to meet you all again, and I hope you have a good
great
season.
Enjoy.
Very much.
Bye.
Okay, take care. Bye-bye.